Coursing through your veins right now is this ancient, incredibly powerful defense system. I mean, we're talking over 500 million years old. It's this cascade of proteins that can spot, tag, and just obliterate invaders with stunning precision. Today, we're going to dive into this absolute masterwork of our innate immunity, the complement system.
So, our story actually kicks off with a mystery, a real head-scratcher for scientists back in the late 1800s. See, they noticed something weird. Fresh animal blood? Incredibly good at killing bacteria, but if you took that same blood and just heated it up a little, poof, that bacteria-killing power completely vanished. So, something in the blood was super sensitive to heat, but what on earth was it?
Well, the mystery started to crack in the 1890s. A researcher, Jules Bordet, he came along and proposed this two-part solution. He said, okay, there's a stable component, which, spoiler alert, we now know are antibodies, and then there's this other mysterious heat-sensitive part. A few years later, the immunologist Paul Ehrlich, he gave this mystery component its name. He called it complement, because it seemed to complement or complete the work of the antibodies, and the name stuck.
So what exactly is the complement system? Well, think of it as a squad of over 30 different proteins, mostly made by your liver, just circulating in your blood. And here's the key. Most of them are totally inactive. They're what we call zymogens, which is just a fancy word for an enzyme that's off and waiting for a signal. But when that alarm finally goes off, they spring into action, activating each other in this incredibly precise, super fast chain reaction.
And honestly, the best way to picture this is as a molecular domino effect. It's perfect. One protein gets knocked over and it triggers the next. But here's the cool part. That one domino then triggers a whole bunch more. So you get this insane amplification, this cascade that turns one tiny little signal into an all-out defensive assault.
All right, before we dive any deeper, we got to talk about the names because, I'm going to be honest, they can be a little confusing. The main proteins are called C1 through C9. But, and this is super important, they're numbered based on when they were discovered, not the order they actually work in. Crazy, right? Now, when these proteins get activated, they split. You get a small A fragment, which usually floats off to act like a flare, calling for backup. And you get a large B fragment, which is the one that physically sticks to the bad guy. The only weirdo in the group is C2, where C2A is actually the large piece. Just a little quirk to keep in mind.
Okay, so this whole molecular domino rally can get started in 3 completely different ways. You can think of them as different tripwires for different types of threats, but they all lead to the exact same destination. So, let's break them down. First up, we have the lectin pathway. This one is ancient. It's all about pattern recognition. See, your blood has these proteins called lectins, and they are basically experts at spotting specific sugar patterns, things like mannose, that you find all over the surface of bacteria and viruses, but crucially, not on your own cells. It's exactly like your immune system recognizing an enemy's uniform. So here's the play-by-play. This protein, mannose-binding lectin, or MBL for short, it physically grabs onto the pathogen's surface. That contact flicks a switch, activating an enzyme attached to it called MASP2. Think of MASP2 as a pair of molecular scissors. It immediately starts snipping 2 other complement proteins, C4 and C2. Then the big pieces that are left, C4b and C2a, they link up right there on the pathogen's surface, and they form this absolutely critical new enzyme, the C3 convertase. You're going to want to remember that name. It's a big deal. All right, next up is the classical pathway. And it's kind of funny. Even though it was discovered first, it's actually the newest one evolutionarily. What's so brilliant about this pathway is that it connects our fast-acting innate complement system with our more specialized adaptive immune system. It usually gets triggered when this big protein complex, C1, latches onto antibodies that have already marked a pathogen for death. It's like the special forces showing up after the spies have already tagged the target.
So how does this one work? Well, when that C1 complex binds, it switches on its own built-in pair of scissors, an enzyme called C1s. And guess what C1s does? You got it. It snips C4 and C2. The big fragments, C4b and C2a, then come together on the pathogen's surface and form, the exact same C4b2a enzyme, the C3 convertase. How about that? 2 totally different triggers, but they build the exact same machine.
And that brings us to our third and final pathway, the alternative pathway. This one is the oldest, maybe the wildest, and you could argue the most important of the 3. It's like a constant surveillance system that's always on, always simmering just below the surface.
It doesn't need a special invitation like the others. It can just spontaneously fire up on microbial surfaces. And this is key. It acts as a massive amplification loop that can turbocharge any complement response.
Yeah, this pathway is totally unique. It gets started with something called tickover. All that means is that the see-through protein, which is everywhere in your blood, just naturally changes shape at a very slow, constant rate. When it does, it can briefly team up with some other proteins, factor B and factor D, and make a really short-lived C3 convertase. This just pops out a few molecules of C3b. Now, here's where it gets cool. If one of those C3b molecules happens to land on a pathogen, bam! It kicks off this powerful, self-sustaining feedback loop, building a different, much more stable C3 convertase called C3bBB.
Okay, so let's recap. We've got 3 different ways to kick things off. The lectin spotting patterns, the classical pathway, seeing antibodies, and the alternative pathway just going off on its own. But all 3 of these roads are leading to the exact same place. They're all racing towards one single critical event. This is the convergence point, the point of no return. And this right here is the whole point. Doesn't matter how they started, doesn't matter which proteins they used. All 3 pathways have one single, unified goal, and that is to build an enzyme on the surface of the invader, an enzyme called a C3 convertase.
And this is where the real explosion happens. C3 is by far the most abundant complement protein we have. And that C3 convertase enzyme, it's like a machine gun just relentlessly chewing through thousands and thousands of C3 molecules. Every time it does, it unleashes C3b, a fragment with this hidden super reactive chemical bond. That bond is like superglue. It lets C3b permanently lock onto the pathogen's surface, basically painting the invader with thousands upon thousands of "eat me" signs.
All right, so now the bad guy is completely covered in these C3b tags. The stage is set and the endgame begins. This is where the complement system unleashes its 3 primary functions. It's a triple threat. It calls for backup, it tags the enemy for disposal, and it launches its own direct lethal attack.
Let's run through this 3-pronged assault. First, recruitment. Those little A fragments we talked about, C3a and C5a, they float away and act like chemical flares, screaming for help and recruiting our trash-eating immune cells, the phagocytes. Second, tagging. That thick coat of C3b on the pathogen, that's called opsonization. It basically makes the invader look delicious to those phagocytes, so they gobble it right up. And third, for the final kill shot, the cascade builds one last devastating weapon, the membrane attack complex, the MAC.
And this is the incredible result. Just look at this. What you're seeing here in this image are the actual holes, the pores, punched into a cell by the membrane attack complex. Each one of those little rings is a molecular drill that has bored a hole straight through the pathogen's outer wall. It's a direct, physical attack that literally causes the invader to pop like a balloon.
So how does it build this thing? It's actually really elegant, step-by-step assembly. It all kicks off when C3b joins the party to create a new enzyme, a C5 convertase. That snips C5, and the C5b piece that's left over becomes the foundation. It grabs C6, then C7, which jams the whole thing into the pathogen's membrane. Then C8 comes in and shoves it in deeper, and that's the trigger. It causes a whole flood of C9 proteins to rush in and snap together, forming that final, deadly pore.
Okay, so this has to be raising a pretty big question in your mind, right? If we have this incredibly powerful, sometimes spontaneously firing weapon just floating around in our blood, how do we keep it from attacking our own healthy cells? I mean, why doesn't the complement system just tear us apart from the inside out?
Well, the answer, in one word, is regulation. A pathogen's surface is basically a perfect landing zone for complement. It has no protection, and its surface chemistry actually helps stabilize the attack. Our cells, on the other hand, are completely different. We have them studded with special regulatory proteins that act like tiny bodyguards, and their whole job is to instantly shut down any complement proteins that accidentally land on them.
And these are some of our star players, our molecular bodyguards. You've got C1 inhibitor, which jumps on the C1 complex and shuts down the classical pathway right at the start. Then you have proteins like DAF, which are like a demolition crew, just tearing apart any C3 convertases that might form on our cells. Factor H and Factor I work as a team to find any stray C3b on our cells and permanently deactivate it. And last but not least, there's protectin. It sits on our cells and puts a cap on the MAC, physically blocking C9 from forming the final hole.
So when you step back and look at the whole thing, the complement system is just an evolutionary masterpiece. It's fast, it's powerful, it amplifies signals like crazy, and it's this beautiful bridge connecting our immediate, innate defenses with the super-specific power of our adaptive immune system.
But of course, a system this powerful doesn't go unchallenged. It's locked in a permanent, high-stakes arms race with everything that tries to invade us. And that leads to a final, really fascinating question. What happens when the pathogens start to fight back? When they develop their own tricks to evade and disarm this system? Well, that's a story for another day.